I have sent you the slides. The first part, you guys are going to skip it because this is related to histology. We're going to do embryology of the blood in 5th lesson of histology. Okay, so when we do blood as a whole, this is when we're going to cover a topic which is called hematopoiesis. Even though it is an embryological topic, it will be covered in histology and asked in histo as well. Okay, so you're going to see questions about it in histo. So we're going to skip all of it. We're going to cover it when we reach our 5th lesson of histology. So we start from this point. We start from the gametogenesis. So first thing first, few words about the embryology. First semester is easy because you're going to see quite a few of the past papers. Okay, so lots of students, what they do, they just take 1 day, they just memorize past papers, they pass, and that's it. The problem arises next semester when you have to talk about these topics in oral exam. So as always, you know my philosophy, just please study it very well this semester so you don't have to talk about it or refresh your memory next semester. Okay, so try to use your 3D vision about these topics and everything is going to be great. So first thing we're going to cover, we're going to look at this picture. As you know, your recommended book is Langman, Embryology. I don't recommend buying that book. I don't recommend reading it. The only thing you need is my notes because this book has lots of mistakes and anatomy department knows about those mistakes. And in the lectures, they typically tell you that this is a mistake, please correct it. Okay, this is a mistake, please correct it. So don't buy the book, don't bother, just focus on my notes and you will be fine. So this embryo that you can see here is 4 weeks old embryo. Okay, in the book it says 3, but it is actually 4 weeks old embryo. What you can see on this little point, this is the head of the embryo. This is where the brain is going to be developing. This is the, pardon my French, ass of the embryo, right? So this is where the anal region will be. This is the back of the embryo. This is where the spinal cord will be. And this region right here is the future anterior abdominal wall. And as you can see on the anterior abdominal wall of the embryo, we have some kind of blister just present there, right? So it's just sitting there, some kind of blister. This blister is known as the yolk sac. And you need to remember that it arises on day 14 of embryological development, but it disappears during the 3rd month of embryological development. As you can see, if you look at your anterior abdominal wall right now, there is no blister. There's just your skin and muscles and that's it. But in embryo, from 2nd week until 3rd month of embryological development, we can see that there's some kind of blister present there. This yolk sac is very important. We'll talk about its functions as we go. Where is the heart of the embryo? We're going to learn about the heart next semester, but you can see that the heart has a very weird position. During the 4th week of embryological development, the heart is sitting in the neck. So our heart develops right here. And next semester, we're going to learn that something will be pushing your heart way down into the thoracic cavity. As a result, all of the vessels which are coming from the heart, they are very long. Look at your carotid arteries, look at your subclavians, they're very elongated. Why? Because the heart was pushed all the way down into your thoracic cavity. But it started to develop in the neck, right? So is it clear until this moment? So head, heart, back, front, ass, etc. Where is the embryo is sitting? This cavity, which is the home of the embryo for 9 months, this is known as the amniotic cavity. And for sure, in lots of movies, you heard the sentence, my water broke, right? They say, my water broke. What does it mean? It means that this amniotic cavity has ruptured. So now all of the amniotic fluid is exposed to the outside. The embryo is ready to be born. This little cord-like structure, as you can see, is connecting to this area of the embryo. This is the future umbilical cord. For now, it has a different name, but this is where the umbilical cord will be. That means that this little tiny dot right here, this is the future belly button, right? So that's where the belly button will be. Is it clear?
Yes.
Okay. The process that we're covering right now is known as the gametogenesis. So production of gametes. Anybody knows what are gametes?
Haploid cells.
Yeah, so these are the sex cells. We have two types of gametes. We have the oocytes in the female, and we have the spermatozoa in the male. They are haploid. What does it mean that they're haploid? It means that they only have one set of chromosomes. So if I look at the oocytes, every single oocyte has only one chromosome inside, and this chromosome is an X chromosome. 100% of the oocytes, all of them, doesn't matter which one are you going to look at, inside of it, you'll find one set of chromosomes. This is going to be the X. What about for spermatozoas? It depends, because 50% of the spermatozoas could have X chromosome, while another 50% of the spermatozoa could have Y chromosome. So the sex of the embryo, the gender of the embryo, what does it depend on?
On the male, on the spermosomes?
It depends on the spermatozoa. It depends which exact spermatozoa has fertilized your oocyte, right? Because as you can see, for oocytes, all of them are X. If spermatozoa carries X as well, and it fertilizes your oocyte, you have a diploid cell, which is now carrying two X's. This is a future girl. If spermatozoa carries Y chromosome and it fertilizes your oocyte, now you have a male because it has X from the oocyte and Y from the spermatozoa. So do you understand the meaning of the haploid and diploid, right? So diploid is XX or XY. Haploid is whether you have X or Y. 100% of the oocyte population have X chromosome only. 50% of spermatozoa have X, 50% have Y. Gametogenesis means production of gametes. It means how exactly are we making oocytes, how exactly are we making spermatozoa. That's what we're going to be covering today, okay? So how can I make an oocyte and how can I make spermatozoas? The problem with embryo, the problem with this course, is that you constantly have to ask yourself two questions. If we're talking about oocyte, for example, right? We have to ask ourselves two questions. Where does it come from? And what does it give rise to? You have to constantly ask yourself these two questions for any embryological structure, right? Where does this thing come from? And what exactly is it going to give rise to? So what is our first question? We have to ask ourselves, where do oocytes come from? Where do spermatozoas come from? Surprisingly, both of these sex cells arise from cells which are called PGCs, primordial germ cells. These cells are very important. They give rise to both gametes. They give rise to the oocyte and to the spermatozoa. The word primordial means before birth. So could you find primordial germ cell in an adult? If you see a question like this, if you see a statement like this in the embryo exam, PGCs are present in adult. Is it correct statement?
False.
It is a false statement, very good, because PGCs are only present in embryo. That means that these cells, these primordial germ cells, if they give rise to the oocytes and to spermatozoas, this process has to happen in embryo. It has to happen during embryological development and not when you're adult. But it's a bit actually also tricky, because in men, as you know, we make spermatozoas constantly. The man will be dying on the bed, okay, but still be able to produce spermatozoas. For female, we are born with a certain number of the oocytes, right? So we can't make them as an adult. So is this point clear? Where do oocytes come from? They come from PGC. Where do spermatozoas come from? They also come from PGCs. Now, how do you know that PGC will give rise to the oocyte? You have to look at its chromosome. PGCs, unlike the gametes that they give rise to, are actually diploid. These cells actually have two sets of chromosomes. Let's imagine that I have a PGC here, and inside of this PGC, I have a diploid set of chromosome. It is an XX. This PGC will be undergoing division. It will have to divide. As a result, it gives rise to two cells now, and each of the cell carries X chromosome. How do you call these cells?
Oocyte.
Very good. You call them oocytes. Why oocytes and not spermatozoa? Because 100% of these cells have X chromosome. So how do I know that PGC will give rise to the oocyte? If it carries X chromosome. Only in that case, PGC will give rise to the oocytes. Let's look at a different scenario. I have a PGC here, a primordial germ cell, and it's not carrying XX, it is carrying XY chromosome. This cell has to divide, it has to split, it has to give rise to two new cells. So I have one cell here and I have one cell here. What will be the chromosomes in these cells?
X in one of them,
X and
Y
Y.
in the other.
X in one, very good. Y in another.
So
you
one
can see that,
oocyte and one sperm cell.
no, you can see that as a population, 50% has X chromosome, 50% has Y chromosome. This is a population of cells. This is a one society of cells. If you look at it, 50 or half of them have X chromosome. 50 or half of them have Y chromosome. What does it describe? Does it describe oocytes or spermatozoas?
Spermatozoas.
Spermatozoas. So do you get what I'm trying to tell you? If PGC has XX chromosomes, the only thing that it can give rise to are oocytes, because when it splits, it gives you two cells. One has X, one has X. That's it, right? If PGC has XY chromosomes, when it splits, one cell has X, one cell has Y. 50% of the cells have X, 50% has Y. What are we describing? We're describing spermatozoa. So the cell that arises from PGC depends on the chromosomes that PGC carries. Okay, this is a very important sentence. The next thing that we need to talk about is, where do PGCs come from? Because you remember what is wrong with the embryology. We constantly have to ask ourselves, where does a thing come from? How old was this embryo?
4 weeks.
4 weeks old. Let's draw an embryo which is 2 weeks old. So 2 weeks before this moment. How do we call the top cavity? Amniotic. Amniotic will always be at the top. How do we call the bottom cavity?
Yolk sac.
Yolk sac. It's always at the bottom. What should be here connecting you to placenta?
Umbilical.
Umbilical cord is right here. In the middle, in this area, this is where the embryo should be, right? This is what we see in this picture when an embryo is 4 weeks old. But this embryo is 2 weeks old, and it only consists of 2 layers. The top layer is called epiblast. The bottom layer is called hypoblast. So an embryo during the 2nd week of development only consists of 2 layers of cells, the top being epiblast, the bottom being hypoblast. Together, these 2 layers are called bilaminar germ disc. So do you see how many changes are happening in just 2 weeks? During the 2nd week of development, your entire embryo is just 2 layers of cells, epiblast, hypoblast, that's it. Literally 2 weeks later, you look at the 4-week-old embryo, it already has a head, it has a heart, it has a back, front, ass, it has all of these structures. So in just 2 weeks, you convert the 2 layers of cells into a human being. The question we were trying to answer, where do PGCs come from? They come from the epiblast during 2nd week. Okay, so PGCs are arising from the epiblast during the 2nd week of embryological development. Let's recap it quickly. What are gametes?
Sex cells.
Sex cells. Sex cells are your oocyte and your spermatozoa. Which chromosomes are present in the oocyte?
XX.
X.
X only. Oocyte is a haploid cell. It cannot have XX. It has to have X only. So 100% of the oocytes, they have 1 chromosome inside, which is an X. Which chromosomes are present in the spermatozoas?
X and Y.
50% can have X, 50% Y.
Exactly. So you cannot choose the option that says X and Y. You have to choose the option that says X or Y. Because if you say that it has X and Y, you're saying that this cell is diploid. But spermatozoas, I repeat, are haploid. So inside of themselves, they can either have X or Y chromosome, right? So 50% of spermatozoas in men have X, 50% have Y. As a result, the sex of the human being, the gender, it depends on the spermatozoa. It depends which chromosome is it carrying. Remind me, where do gametes come from?
From the chromosomal germ cells.
They come from now on, we're saying PGCs, from the primordial germ cells. Primordial germ cells, are they haploid or diploid?
Diploid.
They're diploid. If I want to make oocytes out of the PGCs, which chromosomes should PGC have?
XX.
XX. If I want to make spermatozoas out of the PGCs,
XY.
they should have XY. Is it clear? Where do PGCs come from? Epiblast. When? Second. Now, the area where PGCs become the gametes, this is your future testes or ovaries, right? Because where are my oocytes? They're in the ovaries. Where are my spermatozoas? They're in the testes. So the area where PGCs should convert to become spermatozoa or ovaries, it's there. In embryo, testes or ovaries have a name, and that name is called gonads. Okay, so gonads. Gonads are pairs of organs that could be testes or ovaries, depending on what do you have inside. Okay, so gonads, that's what they are. So even though PGCs arise from the epiblast during the second week, that's not where they are going to stay. They have to move to the gonads. Okay, so they have to go to the gonads. So how are they going to do so? During the second week, PGCs come out from the epiblast. For Professor Antle, next semester, it's very important that you say they detach from the epiblast and start to migrate around the wall of the yolk sac. This is why during 4th week of embryological development, you can see PGCs on the posterior wall of the yolk sac. So it takes them 2 weeks to come from the epiblast, detach, migrate around the wall of the yolk sac, and reach the posterior wall of the yolk sac. How do you know that this is posterior wall and not anterior?
From the head.
Until
The head is on the opposite end. The head is here. That means that this part of the yolk sac is anterior. The tail is here. That means that this is posterior part of the yolk sac. Okay, so try to see the directions. So we said that PGCs come from the epiblast, second week, migrate around the wall of the yolk sac, reach posterior wall of the yolk sac during 4th week. Where are they going? Why are they detaching? Why are they migrating? They are going towards the gonads. Next semester, we're going to learn that gonads are located right here in the lumbar region of the embryonic back. So PGCs are going towards the gonad. How long, in how long will they reach this area? 1 more week. So during the 5th week, they are actually reaching the gonads. So 3 numbers are crucial for you. Second week, PGCs come from the epiblast. 4th week, you can see PGCs in the posterior wall of the yolk sac. 5th week, you can see that PGCs have reached the gonads. Is it clear? There's something called polar bodies, and there's like one question, but I guess it's important. So on this picture, you can see the overview of the spermatogenesis and oogenesis. Without going too much details into this, I want you to understand that in men, everything is great. Every time you see a parent cell, it gives rise to 2 daughter cells, and everything is great. All of these cells are normal, functional, everything is amazing. The problem is in the oogenesis, because every time the oocyte divides, it gives rise to one normal looking oocyte and one oocyte with little to no cytoplasm. You can see that this oocyte is looking a bit small. Why? Because it doesn't have the cytoplasm, or it has, but very little amount of it. So the oocyte, which has little to no cytoplasm, has a name. It is called polar body. I do not want you to confuse it with genetics, because in genetics you also have a polar or bar body. Bar body or polar body in genetics is inactive X chromosome, as you remember. Okay, so it's something different there. This is not that. In embryology, polar body is just the oocyte with little to no cytoplasm. But take a look. Every time the oocyte divides, it gives you one normal oocyte and one polar body. But why polar body is dividing? Do you see? It also gives rise to 2 more polar bodies. How is it capable of division?
Nucleus.
Very good. It still has the nucleus. We said that it has little to no cytoplasm, but is there anything wrong with the nucleus? No, the nucleus is intact and functioning. So in the exam, especially next semester, be careful here. They can trap you and say polar body is inactive. it's not inactive. It's very active because it is capable of division. It does divide and give rise to 2 more polar bodies. So take a look at the oogenesis. We have 1 parent cell and 4 daughter cells, the same as in the male, 1 parent cell, 4 daughter cells. But in male, these 4 daughter cells are functional. They can function properly. In female, only 1 oocyte is functioning because unfortunately, you do need cytoplasm to be a normal looking cell. So in female, every time the oocyte divides, it gives rise to only 1 functioning oocyte and 3 polar bodies. Is it clear? Let's talk about spermatogenesis. I will draw it for you a little bit. Okay, so could you remind me, where do spermatocytes come from? PGCs. And these PGCs are very special because they actually have
Gametes.
And Y.
They have X and Y. They will give rise to spermatozoas, but inside they have X and Y chromosomes. When do the PGCs arise?
In the second week.
Second week. When do they reach the gonads?
4th week.
5th week.
5th week. Because during the 4th week, you can see them at the posterior wall of the yolk sac. What are gonads?
Future ovaries or testes.
It is your future testes or ovaries. In the next semester final oral, Professor Antel will not accept anything unless you say undifferentiated sex organs. Okay, that's his answer. Gonads are undifferentiated sex organs that could become testes or ovaries, depending what you're cooking in them. So how does a gonad look like? How does a gonad in embryo look like? Very easy. It's a circular structure, which is paired. We have 1 on the right, 1 on the left. Think about testes, think about ovaries, right? They're circular paired structures. On the surface of the gonads, we have simple cuboidal epithelium. And this simple cuboidal epithelium is important, especially in the female, because only in the female it has a name. So gonads are present in both, in male and in the female embryo. But only in the female embryo, this surface cuboidal epithelium has a name. It is called germinal epithelium. I think you can see, right, that we have lots of germ, right? Primordial germ cell, bilaminar germ disc, etc. This word is very important in embryo. So I repeat, this is your gonad, 1. You actually have 2. On the surface of this gonad, you have simple cuboidal epithelium, which in female embryo has a name. It is called germinal epithelium. But what is in the core of the gonad? What is inside? What is actually the consistency of the gonad? Well, anything which is anywhere in embryo. How do we call embryonic connective tissue?
Mesenchyme.
Mesenchyme. So inside of the gonad, you can find mesenchyme. This is very important. What is so special about mesenchyme?
Pluripotential stem cells.
Amazing, guys. Pluripotential stem cells. So keep that in mind. Keep in mind that these gonads are actually possessing a bunch of stem cells inside, for sure. So you can see that this is a PGC number 1. There is another PGC somewhere here carrying XY chromosome, and there is another PGC carrying XY chromosome as well. So this is approximately the beginning of the 5th week. So these PGCs have reached your gonads. Now they're ready to invade it. They're going to go inside the gonad, or gonads, right? Because there are 2 of them, I repeat. I hope it's clear that there are 2 of them. And these PGCs, as you can see, carry XY chromosomes. They come into the gonad. And now, very important point. Do they start dividing mitotically or meiotically?
Mitotically.
Very good. they have to start dividing mitotically first. If you are wondering why, very easy. The amount of PGCs which is reaching the gonads is very little. We want to make as many replicas of them as possible to make sure that they can fill up this entire area, so they can fill up the gonads. So I repeat, when PGCs reach the gonads, they invade the gonads and start to divide mitotically. They make exact replicas of themselves. From now on, PGC looks like a dot because I don't have room to draw it, yeah? So you're gonna see that they look like a dot. Do you see this dot? This is a PGC. This is also a PGC. This is also a PGC, yeah? So they look like little tiny dots inside of the gonads. These PGCs inside of the gonads carrying XY chromosomes divide mitotically, but they do not assume a random position. These are males, these are future spermatozoas. They start to align themselves in a very nice manner. They start to align themselves into cords. This is a famous Professor Antol's question. He loves asking this next semester. This question has failed so many students for some reason, but what is the difference between cord and a tubule? How can you know that you're looking at a cord or at a tubule?
Maybe contain vessels.
Eh?
Tubule is empty, but cords are not.
Say better. You're
Like
almost there.
not empty, like they're hollow.
They have a lumen.
Yeah,
Tubules have a lumen. Cords do not have a lumen. Look at my, I don't know, charger cord. If I cut it, you're absolutely right. It is filled with something. There are wires right now inside, right? So it does not have a lumen. It is actually filled with something. But if you look at this pen, you can see that there is a lumen. So technically it is not a cord. Technically it is a tubule. So I repeat that these PGCs, when they come in here into the gonads, when they divide here mitotically, they do not assume a random position. It's not like one goes here, one goes there. No, no, no. They start to nicely align themselves into cords. Okay, so this is one cord. This is another cord that consists of PGCs. Do you get it? So nicely visible cords are present in the gonads of the male. These cords have a very easy name. They're called sex cords. Okay, so these are called sex cords. So sex cords in males start to appear from the 5th week forward. These sex cords are present in the gonads and they consist of PGCs. These PGCs have XY chromosome. Let's imagine the following situation. Let's imagine we aren't talking about a male. We're talking about a female. So I have here a PGC carrying XX chromosome, right? We're in the female. We aren't in a male. The same thing will be happening in the female. These PGCs come into the gonad. They do what? Divide mitotically and also align themselves into the sex cords. But sex cords do not persist in female. They don't. They immediately dissociate into clusters. So I repeat it. In males, sex cords that consist of PGCs stay for some time. In female, they don't. The PGCs align into the sex cords, but then immediately dissociate into little tiny clusters. So the question is why? Why sex cords persist in the male, why they stay in the male, but in the female, they immediately disintegrate into clusters. Professor Rosa will ask you a question differently. She will look at you as a female and she will say, why are you a female? So let's try to answer that. Let's imagine we're next semester, C chance, I'm stressed as hell, and that's the question I get. Why are you a female? So what do you think, guys? Why am I a female?
There's no Y
Very good.
chromosome.
Majority of the students will say because I have XX chromosome. but that's actually a false way to say. It's very sexist, but that's true. I'm a female not because I have XX chromosome. I'm a female because I don't have the Y chromosome. That's why I am a female. On the Y chromosome, if you draw it, this is your Y chromosome. The short arm of the Y chromosome is very special. On the short arm of the Y chromosome, there is a gene right here. And this gene is called SRY gene, or in other words, sex determining region of the Y chromosome. So basically on the Y chromosome, you have the region which is responsible for the sex. This sex determining region of the Y chromosome is coding for a testis determining factor, or TDF. TDF is needed for the sex cords to stay. You need TDF to stay as the sex cords. So let's repeat because it's very confusing. I have PGCs carrying XY chromosome. They come into the gonads during the 5th week. They divide mitotically, but they don't assume a random position, right? They align themselves into the sex cords. To stay in the sex cords, these dots, these PGCs carrying XY chromosome, need TDF. To persist as the sex cord, you have to have TDF. Who can give me TDF? Sex determining region. But where? On the Y chromosome, on the short arm of the Y chromosome. So to paraphrase the question, why sex cords in the female do not persist? What is the simple answer?
Because we don't have TDF, because they don't have TDF.
Because the simple answer is because they don't have the Y chromosome. Okay, the more specific, you're right, because they don't have the TDF, which is on the SRY, which is on the Y. But in simple way, the reason in female the sex cords do not persist because she simply doesn't have a Y chromosome. Is this point clear? Okay. I'm just looking for confusing faces. The confused faces. Can I see confused faces?
Doctor, I have one more question.
Yes.
You know, in PGC, I got like confused. What are these exactly?
They're cells?
Yeah, that produce either XX or XY.
No. You cannot produce XX or XY. XX is a chromosome which is in the nucleus of a cell. So PGC itself is a cell that has a nucleus, and in that nucleus you can have XX or XY. What PGC does give rise to, it gives rise to the oocytes or spermatozoas. Okay, so now we just reached the point that these PGCs are just sitting in the sex cords, and what's going to happen next, right? So tell me, these sex cords persist. So which chromosomes are inside of the PGCs here?
XY.
XY, because the sex cords persist, right? Because of the TDF. Which week was this?
5th.
This is still 5th week, okay? We're still in the 5th week. All of it is happening very fast, but this is still 5th week. In the same time, during the 5th week of the embryological development, you're going to see that some cells of the surface epithelium are going to detach and go into the gonad. So at the same time, during the 5th week, you're going to see that some, not all, some cells of the surface cuboidal epithelium detaches, goes into the gonad and stays. Okay, so it doesn't do anything, just stays. Okay, this was 5th week. When do you think is the next step?
6th week.
That's what you could guess. Anybody else?
7th week.
Not 6th, not 7th week.
9th week, I guess.
No, guys, not even 9th week.
Puberty?
14.
Very good. Somebody said it. The next step is actually at puberty. We don't do anything. Nothing is happening to the sex cords until your boy is born. And not only born, actually reached approximately 12 to 16 years old. Okay, because everybody reaches the puberty differently. Not sure if you know, but we always say in English that I am hitting a puberty, right? I hit the puberty. Why do we say I hit the puberty? Because this process is very sudden, especially in boys, because they go to sleep as a boy, but they wake up as a man. The first symptom that your boy has reached the puberty is that they start to wake up with erections, right? And why do boys have erection? Well, testosterone. Okay, so testosterone starts to flow through the body. So the next step is the puberty. Puberty, unfortunately, is responsible for lots and lots of flowing testosterone in the body. And this testosterone forces your sex cords to acquire a lumen. It forces them to get a lumen. So the presence of the testosterone during puberty forces the sex cords to get a lumen. Can I call them cords?
No, it's the kind of tube.
I can't call them cords because now they have a lumen. So I should call them tubules. The name of these tubules, they're called seminiferous tubules. So a question for you guys. Seminiferous tubules are present at birth, correct?
No, only during
No.
puberty.
Absolutely right. According to anatomy department, it is not present at birth. You're gonna come to the 4th year urology, they are present at birth. Because we have a bit of a testosterone in embryo, we do, okay? How can I convert this embryo into a boy? I need testosterone. So this is why I really love the difference between theoretical years and practical years, yeah? So once you reach the 3rd year, you're gonna realize that so much of the information that we learn in theory years is actually wrong, it's actually useless, okay? So for anatomy department, you're absolutely right, okay? So the seminiferous tubules aren't present at birth. They are gonna be present at puberty. This is why if you palpate, right? So if you touch, if you palpate the testes of a child and you compare these testes to a testes of a male, you will see that the testes of a child are much harder because inside of the testes, there are sex cords. These sex cords do not have a lumen. So as a result, they're like rock solid. But if you palpate the testes of a man, it has seminiferous tubules. Tubules have a lumen, so they're a bit spongy in consistency. So is this clear? Okay, I'm very interested to see what is inside of the seminiferous tubule. Okay, so I really want to see what is inside of the seminiferous tubule. So I take one seminiferous tubule and I do a cross section. I cut it in half. So what are we going to see? So this is a cross section of one seminiferous tubule. There is going to be another seminiferous tubule here, another there, etc. Right? So a bunch of cross sections here. What is this?
Lumen.
Very good. So don't forget, this is a seminiferous tubule. So in the center of that tubule, there is a lumen. Seminiferous tubule consists of a very interesting triangular looking cells. These triangular looking cells are quite new. And if you're thinking that they are PGCs, you're actually wrong because they aren't. These triangular looking cells have a name. You could call them 3 names. All starts with S. Every single name starts with S. You could call it supporting cell. You could call it Sertoli cell. That's the one that we want. And next semester histology, sustentacular cell. Okay, so supporting, Sertoli, or sustentacular cell. Where did this triangular supporting cell come from? Well, you remember during the 5th week, some surface cuboidal cells have detached, went into the gonad and waited. Well, now you can see what did they wait for. They waited to become the Sertoli cells. So these supporting Sertoli cells, they come from the surface epithelium of the gonad. It's very helpful. Sertoli cell comes from surface epithelium. Okay, that's where they come from. Inside of the gonad, we had a bunch of PGCs aligned into the sex cords, as you remember. So where are these PGCs now? Well, PGCs were in sex cords. This is not sex cords. This is seminiferous tubules. So new place, new me, new name. PGCs changed their name, and now they're going to be called spermatogonia. Spermatogonia. So I repeat, PGCs are called so in embryo. In boys, in adults, they change their name. It's exactly the same cell, but it just changes its name, and now it is called spermatogonia. Where these spermatogonia are sitting? In between Sertoli cells. So this is spermatogonia cell here. This is another spermatogonium. This is another spermatogonium, and another one is here, another one is here, and you get the idea. Yeah, so between the Sertoli cells, spermatogonia are sitting. So again, spermatogonia are present at birth, correct?
No.
No, okay, because spermatogonia are sitting in the seminiferous tubules. I don't have seminiferous tubules at birth. I cannot have spermatogonia at birth as well. Is it clear that spermatogonia come from PGC? They are basically
No.
PGC, right?
One more thing. All of this is only for the boys, only.
Yes, we're talking about boys right now because it's a XY, because this PGC has XY. As a female, I cannot have the Y chromosome for sure. Remind me, where do PGCs come from?
Epiblast.
Epiblast.
Epiblast, very good. So again, we have spermatogonia in the seminiferous tubules. These spermatogonia come from PGCs. PGCs come from the epiblast.
Sorry, excuse me. So technically the sex of the baby can be determined by the 2nd week when PGCs are formed by the epiblast?
Technically, but we don't have the instruments to do so. We
Okay.
don't have it. Yeah, we don't have it. We wait for the external genitalia to appear. So after 3rd month, okay? So technically, you're absolutely right. But we don't have the technology for that. Okay, so here are my spermatogonia. They're sitting here. What are these spermatogonia going to do? They have to give rise to the spermatocyte. So how can they do so? Spermatogonia, starting from puberty, starts to divide and gives rise to the primary spermatocyte. Primary spermatocyte divides, gives rise to secondary spermatocyte. Secondary spermatocyte divides and gives rise to spermatid. Spermatid does not divide, it simply matures to become spermatozoa. Notice that my spermatozoas are in the lumen, right? So you can see that spermatogenesis, production of spermatozoa, happens between 2 Sertoli cells. That's why they're called supporting, because they support spermatogenesis physically, nutritionally, emotionally, whatever way, okay? So they're gonna be supporting this entire process from A to Z. So I repeat, again, we have 2 Sertoli cells here. Where did these Sertoli cells come from?
The cuboidal epithelium.
The surface cuboidal epithelium of the gonad, yeah. Why is it called supporting cell? Because it supports spermatogenesis, because it supports spermatogenesis. What are the stages of spermatogenesis? We start with
Spermatocyte.
spermatogonia gives rise to
Spermatocyte.
Primary
primary
spermatocyte,
spermatocyte gives rise to
secondary
secondary
spermatocyte,
spermatocyte gives rise to
spermatid.
spermatid. Does spermatid divide to become spermatozoa?
No, it
No.
matures.
Mature.
It matures. So please remember that. It's very important for the exam. Spermatids are considered to be immature spermatozoa. Okay, that's what they are. They're immature spermatozoas.
Wait, what do you mean by matured and unmatured?
Mature, it means it has a function. Immature, it means it doesn't have a function. In other words, spermatozoa is adult cell. Spermatid is a baby cell. Mature, immature. We have a third cell. We have Sertoli. We have all of these cells here, and we have the third cell. The third cell is not part of the seminiferous tubule. It is actually located between seminiferous tubules. So this is one seminiferous tubule, this is another. So between them, you can see this cell right here. It's between seminiferous tubules. This is known as the Leydig cell, or another name for Leydig cell. It is also known as the interstitial cell because it is in the interstitium, right? It's not part of the seminiferous tubules. It's simply in the connective tissue between seminiferous tubules. Okay, so this is a Leydig cell. What do you think? Where did the Leydig cell come from? We used surface epithelium to make Sertoli cells. We used PGCs in the sex cords to give rise to spermatogonia. What we haven't used? Mesenchyme? Mesenchyme. So Leydig
Mesenchyme.
cells are arising from the mesenchymal stem cells. Okay, they're coming from the pluripotential stem cells. That's where they come from. Leydig cells
So
produce
how do you spell it?
testosterone. Sorry?
How do you spell it? The Leydig cells.
Again?
How do you spell it? How do you spell it? The
How do you
Leydig
spell it? Sorry. Leydig. Leydig. Leydig cells, they come from the mesenchyme, they produce testosterone. A few words about the Leydig cells. They are the first cells to appear. So even before spermatogonia, even before Sertoli cells, they're one of the first cells to appear because they make testosterone. Testosterone is needed to make male sexual characteristics in embryo. I mean, I need testosterone to grow a penis. Okay, so this is why Leydig cell is the first cell to actually arise. Okay, we talked about all of these parts. Take a look at the steps of the spermatogenesis. We have spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, and the last one is spermatozoa. So these are the 5 stages of the spermatogenesis. But in addition to spermatogenesis, we also have spermiogenesis. Spermiogenesis is considered to be the last step of the spermatogenesis. In other words, spermiogenesis is simply maturation of spermatid to become spermatozoa. We said that this cell does not divide, it simply matures. So this process is known as the spermiogenesis. I repeat, for the exam, it's very important. You will see something like this. Spermiogenesis is the last step of spermatogenesis. Correct. Okay, because literally that's what happens. Spermatid matures to become spermatozoa. Here I wrote to you that we actually have 2 types of spermatogonia. We have the type A and the type B. Type A is known as the light cell. Type B is known as the dark cell. Okay, light, dark cell. The beauty of being a male from the reproduction point of view is that you can produce spermatozoas non-stop. Okay, you can make them even when you're 70 years old. They might be very immotile spermatozoas, but they still are there. You still are producing them, okay? So how is it possible that a male, even in his 70s, can still make spermatozoas? The answer lies in a stem cell. For sure, a male has to have a stem cell that is just sitting there and actively dividing. So you need to remember that type A spermatogonial cell will always be a stem cell. That's all it is. This type A stem cell gives rise to type B, and lets type B divide to become primary, then secondary, then spermatid, etc. Type A is always a stem cell. It only gives rise to type B. Let's type B do everything else. Imagine what would have happened if I didn't have type A, if I only had type B. What would have happened? I would have eventually made spermatozoa, right? A male would ejaculate and that's it. You can't make more because there is no type A, there is no stem cell. Okay, so as you can see, that's not the case. The reason you can make more spermatozoa is definitely because of the type A. Very important question that you're going to see not only this semester, but also next semester histology. Spermatozoa is haploid. PGC that gives rise to spermatogonia is diploid. So which cells are which? You need to remember that primary spermatocyte is also diploid. It also has XY. But starting from the secondary spermatocyte, these cells are haploid. Okay, so second spermatocyte, haploid. Spermatid, haploid. Spermatozoa is a haploid cell. Okay, there is something which is called Sertoli cell barrier, or another name for it, it is also known as the blood testis barrier. Blood testis barrier is done to separate diploid cells from haploid. So it is actually located between the primary and secondary spermatocyte. You can imagine it something like this. I have a Sertoli cell here, and I have another Sertoli cell here. Which cell is here? Spermatogonia. This one will be
Primary
spermatocyte,
secondary
secondary
sperma.
spermatid, and spermatozoa. Am I right? So what is this blood testis barrier? This is why they don't like to use the word barrier. Barrier implies that it has lots of layers. Yeah, like your blood-air barrier is 4 layers. Your blood-thymus barrier is 6. Your blood-placental layer is like 8 layers. Okay, there's so many layers there is. They don't like to say that it is a blood testis barrier because it consists of one thing. And that one thing is a tight junction between 2 Sertoli cells. You're going to see that this is a Sertoli cell here. This is also a Sertoli cell, and they're just holding hands right here. And as they're holding hands, this creates the tight junction, right? So their plasma membranes are touching each other in these areas. So that creates the tight junction. Above the tight junction, we can see the diploid cells. Below, we can see the haploid cells. The question is, this junction, this blood testis barrier, who is it protecting? Is it protecting the diploid cells or the haploid ones?
Haploid.
Very good. So blood testis barrier, the Sertoli cell barrier, it protects the haploid cells. From who?
From lymphocytes.
Very good. From immune cells, from lymphocytes. Haploid cells are weird for our body. How come you don't have another chromosome? Like, what's wrong with you? Okay, so that's what is going on through the lymphocytes' mind. So when they see a haploid cell, they don't just kill it, they want to modify it. They want to make it diploid, which is worse. Okay, you don't just kill, you actually experiment on that cell and make it diploid. So think about it this way. These two are the Sertoli cells. This is where the basement membrane will be, right? Here would be the connective tissue. It will have a blood vessel. In that blood vessel, I can have lymphocytes coming out. What do? They see diploid cells. Everything is normal. below the tight junction, they cannot see anything. If I cannot see, it's not there for lymphocytes, okay? So they don't care. So we're trying to protect the haploid cells. Okay, speaking of spermiogenesis, what is happening? So this is what you're doing, okay? I will describe it on this picture. This picture will for sure come up, okay? For sure, they will bring this picture and ask, what do you see? So spermatozoa is approximately, we have to check the lectures this semester because they always freaking change it. But as far as I remember, last year it was 60 micrometers. Okay, so 60. We have to check the lectures. This is going to be your homework. Okay, so you need to open the lecture and see which number did they put for the length. I gave up trying to memorize them because every year it's different. Okay, so 60 micrometers. So this is the length of the spermatozoa. Spermatozoa consist of the head. In our university, we also have a neck. This is neck region. It consists of the middle piece. Until here, it will be principal piece. And then this will be the tail. Principal piece and the tail together are flagellum. And hopefully you remember our first histo self-control test. Flagellum is the longest cilium, right? It is super, super long cilia. So it's quite motile. It has 9+2 axonemal arrangement, right? So hopefully you remember our first histo self-control test. In the middle piece of the spermatozoa, we have a bunch of mitochondria. Mitochondria are present actually everywhere except the head region. In the head, you will not find a single mitochondria. This is why mitochondrial DNA is not of the maternal, sorry, it's not of the paternal inheritance, it's of the maternal, right? But nowadays, they're actually going to change it because there are more and more families being discovered where they actually have paternal mitochondrial DNA. So the spermatozoas in their families have mitochondria in the head of spermatozoa. Okay, so but it's not important for exam. Mitochondria are only present in the middle piece, principal piece, and the tail, right? Head and neck will not have them. What is the purpose of the mitochondria here? This is the little engine of the spermatozoa. Okay, so that's the engine. So it makes a shit ton of ATP. This ATP is being used for the flagellum to do this. Okay, so to do the movement. Head is more important for us because this is where the nucleus will be. This nucleus will be covered by a region which is called acrosome. Acrosome covers only 3/4 of the head. It doesn't cover the entire region, okay? It covers like this. It covers the 3/4 of the head of spermatozoa. Acrosome consists of 2 layers. You don't have to know which kind of layers, just that they're glycoprotein layers. Behind the acrosome, or in the book they say within the acrosome, right here, we're going to have digestive enzymes. And these digestive enzymes are needed to penetrate the wall of the oocyte. So this acrosome has a very important job. Okay, number 1, it actually protects a bit of the nucleus. And number 2, it's needed to penetrate through the wall of the oocyte. Okay, so it possesses some kind of digestive enzymes. Very important. How long can spermatozoa survive in the female reproductive tract? Approximately 3 to 5 days. Okay, so highly tested. How long does spermatogenesis takes? 74 days. Okay, so from spermatogonia until spermatozoa, it takes 74 days. Primary spermatocytes, when they become secondary spermatocytes, are entering super, super long prophase. It is so long that it is actually 22 days. Okay, so to convert our diploid cell to haploid cell, we actually need 22 days. Okay. I think it's important to mention this. The fluid that the man ejaculates, this fluid is called semen. What does the semen consist of? Well, of course, spermatozoas, but also seminal plasma. The fluid itself, the liquidy part of the semen, this is known as the seminal plasma. Anybody knows who makes this seminal plasma?
Zinc maybe, contains zinc.
Prostate. Okay, prostate, the most important gland in the male. Okay, prostate. This is what it does. It secretes this seminal plasma. Anybody knows where is prostate?
It's under the bladder.
Very good. It's actually under the bladder, surrounding the urethra, right? So that's why with age, men have difficulties with urinating because it gets bigger, it compresses the urethra, you can't pee. The question that they will ask you is, what is the composition of the seminal plasma? What does it actually have? Most important thing, it has fructose. I don't think I'm opening America for you here or inventing America, but you know that the energy source for spermatozoa is not glucose, it's fructose. It's the only cell in the body that doesn't use glucose and uses fructose. So seminal plasma contains a bunch of fructose. It contains vitamin C. This is why in some countries they use semen as a beauty mask to whiten the face of a person. Okay, so they make a cream out of the semen. It also has alkalase. Why do we need alkalase?
Survive in the acidic environment.
Because female reproductive tract is actually acidic. For the semen to actually survive in the vagina and uterus of the female, you need to kind of increase the pH, right? So that's what the alkalase is going to be doing. Most importantly, it also has prostaglandins. Anybody knows what is the purpose? There's so many of them, but you don't have to know which one exactly, just prostaglandins. What
I know that they can make like pains.
Yes, they can make pain, but what is the pain is from in this particular situation? Prostaglandin in this particular situation causes smooth muscle contraction. And guess who has lots of smooth muscle? Vagina and uterus. Basically, semen contains a substance that makes vagina and uterus contract. If vagina and uterus contract, they basically bring the semen closer to the oocyte. So this little substance is the one that they're going to test you on. Prostaglandin causes smooth muscle contraction in the female reproductive tract, causing it to make sure that semen can reach the oocyte. Is it clear? Okay, was it difficult? Because now it's going to be difficult. Okay, now it's actually going to be difficult. So if you thought that male was difficult, hold on. Okay, we females aren't difficult because we want to be. It's actually in our genes to be difficult. Everything in the female reproductive system is difficult. Okay, it's super confusing. So try to keep up. So let's imagine we're in the female. So 5th week, what do we have during the 5th week in the female reproductive tract? Well, the same thing, we have the gonads. These gonads consist of
cuboidal surface cells.
They have the surface cuboidal epithelium, the gonads. How do we call it in female?
Germinal epithelium?
We call it germinal epithelium. Why do we call it germinal epithelium? Because many years ago, as you know, from the medical perspective, female was inferior to a man. She could not possibly have the same origin to structures as a man. So they realized that PGCs in the man come from epiblast. In women, they cannot come from epiblast because that means that women and men are equal, and it could not be. Okay, so they realized or made a discovery that PGCs in the female actually come from surface epithelium of the gonad. As a result, this epithelium got the name germinal epithelium. Is this true right now? No. As you know, PGCs, doesn't matter male or female, they come from epiblast, but the name stuck, it got stuck. And unfortunately, especially in the third world countries like Hungary, they still, they still make you learn that, okay, that this epithelium is known as the germinal epithelium. But again, it is known as the germinal epithelium because 150 years ago, they thought female PGCs would come from here and not from epiblast, but you know that that's not true. The core of the gonad consists of the mesenchyme, the same as in the man. So 5th week, we have PGCs. The problem is that these PGCs do not carry XY chromosome. They carry XX because we have to make the oocytes. So what do these PGCs do? They come into the gonad and divide mitotically. As they divide mitotically, they align themselves into
Column and then dissociate.
They
clusters.
align themselves into the sex cords, but because female lacks the Y chromosome, there is no SRY. SRY isn't making TDF, there is no TDF. So you're gonna see that the sex cords dissociate. They dissociate into clusters. So the same idea, every dot represents the PGC. So I have here a bunch of PGCs in one cluster. I have another cluster of PGCs here. I have another one here, and etc. Do you get it, right? So they aren't sex cords, they are clusters of PGCs. These clusters are going to be surrounded by some kind of cells. These cells also come from the surface epithelium of the gonad. They detach, they come into the gonad and literally start to surround the clusters. These cells that are now acting as a border of the clusters are known as the follicular cells. So again, if I magnify into this area, I have a bunch of PGCs sitting in the clusters, right? The dots are PGCs. This cluster is surrounded by follicular cells. Where did follicular cells come from? Surface epithelium. In this semester and next semester final oral, expect a question like this. Which cell is the analog to follicular cell? Which cell is analogous to follicular?
The supporting cells.
Sertoli cell, very good. Supporting Sertoli cell in men. They are coming from the same thing. The only difference is that they're in a man or in a woman, right? So follicular cell is very much similar to Sertoli cell. They both come from the surface epithelium. The only difference is that whether you're a man or female. Okay, what do you think? What is the next step? The next step is that these PGCs during 5th week already start to differentiate into oogonia. In men, we waited until the puberty. In female, we're already late. Okay, so we need to do some additional things. Okay, so you're going to see that these PGCs, they start to divide to become oogonia. Oogonia should divide to become primary oocytes. Primary oocyte should divide to become secondary oocyte. But let me draw it here for you. Okay, so let's imagine that we have one cluster. In this cluster, okay, I'll draw it here. So 5th week, we're looking at one cluster. In this one cluster, I have PGC, I have PGC, I have PGC. Okay, let's imagine 4 PGCs are sitting. In female, we start the process of oogenesis immediately. So during the 5th week already they start to divide. But to kind of help you, I want you to see what happens throughout time. Okay? So first thing that we're going to do, you're going to see that we're going to look at the 4 month. So the same cluster, but what are the changes in that cluster during the 4th month? You're going to see that some oogonias stay as oogonia, no changes, procrastinators, okay, nothing is happening. So 3 months have passed, 0 changes. Some oogonia has already differentiated to become, some PGC has already differentiated to become the oogonia, which is normal. Some PGC is like a headless chicken, already became oogonia, and now is actually a primary oocyte. And some PGCs stay as PGC. So I repeat so you don't get confused. Which cells are these?
PGCs.
And they're sitting in one cluster during the 5th week of the development. We're looking at what happens to these cells throughout time in pregnancy, in embryo, yeah? So what happens to them throughout embryological development? So you look at the same cluster, but this is already 4th month of the development. Which cells can be seen in the cluster during 4th month?
Arconium, primary, oocyte,
PGCs.
So PGC, oogonia, and primary oocyte. Basically all of them except for the secondary oocyte. You need to remember that primary oocyte becomes secondary oocyte at puberty. This is 4 months old embryo. Would you ever see secondary oocytes in embryo? Never. Okay, the maximum you can see is the primary oocyte. Let's look at the same cluster 1 month later. So 5 months old embryo. If you were a primary oocyte, you do nothing. You stay as the primary oocyte. If you were the oogonia, you simply become primary oocyte. That's it. But if you were the PGC that was procrastinating for 3 months, now you have to quickly hurry the hell up to become quickly oogonia, so you can quickly become primary oocytes. So do you see what's happening in the cluster during the 5th month? We can only see primary oocytes. Again, which cells can you see in the cluster during 5th week?
PGC.
PGCs only. Which cells can you see in the cluster during 5th month?
The primary oocytes.
Oocytes only. Which cells can you see in the cluster during 4th month?
Arconium,
Primary oocyte PGC, and what's this?
Oogonia. Okay, so you can see PGC, you can see oogonia, and you can see primary oocyte. Are the steps of the oogenesis are clear or not really? Because it's not very difficult. Yeah, so we have PGC. PGC should give rise to the oogonia. Oogonia should give rise to the primary oocyte. Primary oocyte should give rise to the secondary oocyte. Just this step is in puberty. This step is in embryo. Is it clear?
When is puberty exactly?
It's very it's very interesting question because it depends. Okay, so in girls normally from 10 to 15, meaning that if you had your menstruation at 9, it's abnormal. If you had it at 16, it's also abnormal. So 10 to 15, that's when you hit the puberty as a girl. Okay, I want to count how many primary oocytes have I made in both ovaries during 5th month of development. Okay, both ovaries. embryo,
8.
5 months old. How many primary oocytes are in
8.
both ovaries?
8.
8, that's it?
Like in what you draw, it's 8.
Yeah, but I can't draw all of them, can I? Like, you know, it's just a sympathetic, schematic representation here. But yeah,
By 40 by 12, right?
I will help you because you aren't even close. In both
That means-
ovaries, during 5th month of embryological development, in clusters, if you count every single primary oocyte, you will count 7 million primary oocytes. So 3.5 million in one ovary, 3.5 million in another ovary. I really want you to really focus on that. 7 million primary oocytes. All this primary oocyte has to do, it has to become secondary oocyte. Secondary oocyte has to get fertilized. You have a human being. You have potential 7 million people in 2 ovaries, in one girl. Hungary is 10 million people. One woman hypothetically can reproduce the entire Hungary. So I'm trying to tell you that 7 million primary oocytes is too much. Agree with me? That's too many people in one female ovary. Again, it's not a person there, it's half of a person. Yeah, because oocytes, spermatozoa. So apoptosis begins. We got too excited, we made too many primary oocytes. Now we need to kill them off. And I really like how it happens. So this is the gonad. In this gonad, we have a bunch of clusters. What we have just saw, right? Let's imagine in this cluster, I have 100 primary oocytes. 99 will die. In this cluster, I have 200 primary oocytes. 199 will die. In this cluster, I have 10 primary oocytes. How many will die?
9.
9.
9.
So do you see what's happening?
Yeah.
It doesn't matter the amount of primary oocytes you have in the cluster. Only one should remain. So apoptosis in the clusters begins at 5th month and it is done at 7th month of embryological development. So how many primary oocytes should I have right now? 7th month in the cluster, sorry.
1.
1. There should be only 1 primary oocyte left. So we had 4 primary oocytes in our cluster. So by the time you reach 7th month of embryological development, 3 of them should die, only 1 of them should stay. Can I call it a cluster if there's only 1 primary oocyte? No. So what should I call it? What surrounds this primary oocyte?
Follicular.
Very good. Follicular cell surrounds it. You remember what was surrounding the clusters? This is a cluster which had a bunch of cells in it. What was surrounding the cluster? We had follicular cells. Yes, we killed majority of the primary cells in this cluster, but follicular cells persist. So what is present outside of your primary oocyte are follicular cells. So how do we call this structure?
Follicle?
Follicle. Follicle in simple English is egg. Whenever I say, oh, my eggs are dying. I'm 28, my eggs are dying. So what's happening? My follicles are dying, right? My primary oocytes surrounded by the follicular cells are dying. Is it clear? Are you going to be using these follicles anytime soon? Are you going to use this primary oocyte anytime soon?
No.
Your 7th month old embryo. When are you going to be using this primary oocyte?
When we hit puberty.
Puberty. So what do we need to do? We need to freeze this primary oocyte until the puberty. How we do so? Follicular cell starts to secrete a substance. This substance is known as the oomy. OMI stands for oocyte maturation inhibitor. Okay, so this substance, OMI, it is secreted by the follicular cells. What does OMI do? Starting from 7th month of embryological development, you're going to see that primary oocyte enters meiosis 1 to become secondary oocyte. So during the 7th month of embryological development, we already start the production of secondary oocyte. We allow primary oocyte to enter meiosis 1 to become secondary oocyte. The problem is that if this OMI is present, primary oocyte is arrested in the first phase of the meiosis 1. The first phase is called prophase. OMI arrests primary oocyte in the prophase. It doesn't allow it to become secondary oocyte. Only during puberty, this OMI will stop its production. It will not be there. If there is no OMI, primary oocyte can continue to become secondary oocyte. So how do we freeze the oocytes for later use? I repeat, we allow primary oocytes to enter meiosis 1, but we arrest it at the prophase. What arrests it at the prophase? The OMI. Who makes the OMI? Follicular cell. The cell that surrounds the primary oocyte will be secreting the OMI. OMI keeps it at the prophase. It doesn't allow it to become secondary oocyte. For sure, you know that the older you get, the more chances that if you get pregnant, you will have a Down syndrome baby, right? So for sure, you have heard that. Down syndrome babies, as you know, they are triplody 21. They have 3 chromosomes 21. Right, so if I'm a girl, I'm gonna have 3 of them there. Could you tell me why the chances of having a genetical disorder are higher if the maternal age increases?
Because, like, as you said, that they will stop, or like they will freeze on the prophase. So, like, for example, radiation will can hit them, can hit the chromosomes.
Because the follicular cell is not working well anymore when you age.
So if it isn't working well, then what happens? It simply stops making the OMI, and your primary oocyte becomes secondary. Secondary gets fertilized, and that's it, you have a baby. But I think Magdi was kind of going there. It's just that you added the radiation part, okay? That's not why, okay? Not the radiation. Take a look. This is 7th month old embryo. You have a 40-year-old female coming to you and saying that she wants to get pregnant. What do you think? The follicles that she has, are they young?
No.
No, they're 40 years plus 7th month. Okay, so they're actually like 41 years old. And the follicles that she has for 40 years are in the prophase. They aren't just a primary oocytes. They're already in the process of becoming secondary. There is something cooking in the nucleus for 40 years. They're trying to divide to become secondary, but OMI isn't allowing. So what do you think? You're messing with the nucleus of a primary oocyte for 40 years. Do you think you're gonna have a healthy baby? Well, 1 out of, 100, okay, I will rephrase it because it's very difficult to say. Basically, the probability that at the 40-year-old you'll have a Down syndrome baby is 1 out of 100, which is actually quite high if you think about it. Because as a 17-year-old, it's 1 out of 10,000.
But 25 years as well is a long time. So why is this okay?
But I still have more follicles. We're going to see right now. By the time you're 40, you're approaching menopause. Okay, so slowly you're approaching menopause. Menopause is when you have no follicles whatsoever, right? So you have no oocytes whatsoever. So when you try to get pregnant at 40 years old, it's very difficult to get a healthy baby because all the follicles which are left aren't that good. The best follicles are until you're 30 years old. That's the best ones. After 30, they're okay. Okay, so the best ones are until 30. Yeah. Okay, so yeah, I should add one more detail. So how do we long? Agree with me, this prophase is going to be very long, right? Because it's until the puberty. So this prolonged prophase, it actually has a name. It is known as the diplotene stage. Okay, so diplotene. So to freeze the oocyte, we put it into diplotene stage. Okay, guys, so I am going to do what? I'm going to continue. I'm going to kill you a bit more. Okay, let's imagine that we finally reach the puberty, guys. Okay, so I'm going to go to this. No, I can't go to this. Should have added one. Okay, doesn't matter. Sorry, guys. Let's imagine that we reach the puberty. Okay, so you have a 12-year-old female. Which gland initiates the whole thing? This gland is known as the hypothalamus. So your entire puberty is controlled by hypothalamus. Hypothalamus secretes a hormone, and this hormone is known as the GnRH. GnRH, known as the gonadotropin releasing hormone. So hypothalamus is going to be secreting a bunch of the gonadotropin releasing hormone. This hormone is going to be working on pituitary gland. These words mean nothing to you. We're going to talk about it next semester, but unfortunately they ask it this semester. Okay, so hypothalamus secretes gonadotropin releasing hormone. This hormone works on the anterior pituitary gland. What is this hormone asking you to do? It is asking you to release gonadotropins, right? Gonadotropin releasing hormone. It is asking you to release gonadotropins. So this is exactly what it does. This pituitary, this is exactly what it does. It releases FSH and LH. It releases two hormones, FSH and LH. FSH stands for follicle stimulating hormone. LH stands for luteinizing hormone. This is happening at puberty. Okay, hypothalamus secretes GnRH. GnRH works on anterior pituitary gland. This gland secretes FSH, LH. What do you think FSH does? Follicle stimulating hormone. What does it do?
It like
Grow more follicles?
It makes them grow. Okay, let's say that. It makes them grow. We haven't talked about these numbers, I just realized. I apologize. Tell me, how many primary oocytes have we had during 5th month of embryological development?
7 million.
7 million.
7 million. How many primary oocytes are you going to have when you're born?
700,000.
600 to 800,000. How many primary oocytes do you have when you start the puberty? 40,000. So when you're born, you have 600,000 primary oocytes, but when you start the puberty, you have 40,000. Why they died, right? So why so much death happens? The reason for that is this OMI. Who secretes OMI? Follicular cell surrounding the primary oocyte. Follicular cell is very random cell. Today it secretes OMI, tomorrow it doesn't. This process is not controlled, it's uncontrolled. It starts to secrete OMI, but it doesn't keep on secreting it. It can secrete it for 40 years or for 40 days. You can't know. Let's imagine I have a follicle, primary oocyte is under the influence of the OMI. Suddenly, no OMI. Follicular cell stops secreting OMI. What does this primary oocyte wants to do now?
Become secondary.
But you're a child. Can it happen in children that primary oocyte becomes secondary?
No.
It can't. Why not? What do we need for a primary oocyte to become secondary oocyte?
You said that
FSH.
We need FSH and we need estrogen. As a child, do I have estrogen? No. So now you have an oocyte in a child that is not under the influence of AMI. It wants to become secondary oocyte, but it can't because there is no estrogen. So what is left to do? Apoptosis. Okay, this is why the number reduces so much, right? You see, when you're born, you have 600 to 800,000 primary oocytes, but when you start the puberty, you have only 40,000 because primary oocytes keep on dying. Okay, that's the problem in a female. In men, we keep on making. In female, we keep on dying, the primary oocytes. Okay, so we keep on killing them off. But guys, again, this is anatomy department. This number is not true. Okay, when you reach the 4th year, you're gonna learn it's more like 240,000. You start puberty with 240,000. Why? Think about it this way. I'm horrible with math, but maybe somebody can calculate it for me. I start the puberty with 40,000 primary oocytes. How many do I choose monthly?
1.
That's how many will mature. But as a girl, I need a choice. So how many do I choose? My options. Actually, 15 to 20. Every month, you choose 15 to 20 primary oocytes, but only one of them will mature. What's going to happen to the rest of them? I chose 15. Only one of them matures. What's going to happen to the rest of them?
Apoptosis.
Apoptosis, they die. So do you see what is the problem with a female? Like, death follows us everywhere. Okay, poor oocytes keep on dying. Like, that's all they do. Since the moment that you made them, they keep on dying. Okay, 7 million, too much. Okay, so we're gonna kill them off. Eventually you're left with like 40,000. Still they have to die. Okay, still every month I choose 15 to 20, but only one of them stays. Can it be 2? I chose 15 to 20. Can 2 mature?
For twin.
Yeah.
Yes.
Yes. How do we call these twins?
Twin.
Paternal.
Non-identical, fraternal. Yeah, non-identical because they come from 2 oocytes, right? Identical twins, they look the same, they come from 1 oocyte, but we'll talk about it at the end of our course. So is it clear, these numbers? Okay, so if you do the math, if according to anatomy department we have 40,000, then it will finish when I'm 30. You will reach menopause when you're 30. That's not the case. You reach menopause when all of the oocytes are finished, when you're about 40 to 50 years old. Okay, so sometime that. So the initial number when you start the puberty should be 240. But again, for the exam, it's 40.
I have a question.
Yeah.
You mentioned about the follicle. You mentioned that.
Yes, follicle is the oocyte, primary oocyte surrounded by the follicular cell.
And you said that females have only one?
No, they had 600 to 800 follicles, 1,000 follicles when you're born, 40,000 follicles when you're starting the puberty. So
they are oocytes, the 40,000 oocytes.
But follicle is primary oocyte surrounded by follicular cell. It's the same thing. Follicle is primary oocyte. Okay, let's start the puberty. We said this: GnRH, pituitary, FSH, LH. I have 2 ovaries. Every month in one ovary, initially I had 20,000 follicles here, 20,000 follicles here, right? Left and right ovary. Every month out of the 20,000 or 40,000 in total follicles, you choose 15 to 20. How can you do so? You have to work with the FSH. Every month, let's imagine in this ovary, I have 20,000 follicles. Randomly, every month, 15 to 20 follicles raise their hand. They raise the receptors for FSH. So when FSH comes in, it only stimulates these follicles to grow. Is it clear? So how do I choose out of 40,000, 15 to 20 follicles? FSH. These 15 to 20 follicles, they raise their hand, they have their receptor for FSH. So let's imagine, let's follow what happens to this follicle throughout. The follicles that we have made in embryo during the 7th month, these follicles are called primordial follicles. You can see that primordial follicle consists of the primary oocyte surrounded by the squamous follicular cells, right? This primordial follicle is going to be raising a hand, which is the receptor for FSH. And 15 to 20 of these primordial follicles raise their receptors for FSH. So FSH comes in, it binds to it, and it starts to stimulate this follicle to grow. Primordial follicle, under the influence of the FSH, starts to grow to become primary follicle. This is a mistake in the book. This is a primary follicle. This is the first stage of primary follicle. This is the second stage of primary follicle. But before we talk about it, take a look. What is the difference between primary follicle and primordial? Inside, you still have primary oocyte. This is its nucleus, this is its cytoplasm. But take a look, the follicular cell has changed its shape, right? Under the influence of the FSH, the flat follicular cell becomes cuboidal. In the later stages, what happens to the follicular cells? They aren't simple cuboidal, they start to proliferate and they become stratified cuboidal follicular cells. What is inside? Still primary oocyte. So this is the nucleus, this is the cytoplasm of the primary oocyte. So I repeat, how many primordial follicles do we choose every month starting from puberty?
15 to 20.
15 to 20 out of the 40,000. Which hormone helps me choose?
FSH.
For these 15 to 20 follicles, they raise their hand, they raise the receptor for FSH. FSH comes in, stimulates this primordial follicle to become primary. What is the major difference between them?
The cuboidal follicle cells grow, or
No.
flat epithelial change to
From
Flat
flat, they become
follicular
cuboidal.
cells grow to become cuboidal, right? So flat follicular of the primordial follicle grow to become cuboidal. They grow even more to become stratified cuboidal follicular cells. These stratified cuboidal follicular cells, as you can see, they look like little tiny dots. They look like little tiny granules. As a result, stratified cuboidal follicular cells have a name. They're also known as the granulosa cells. These cells right here, they are granulosa cells. Granulosa cells, as you can see, appear at the primary follicle stage. Granulosa cells have a very important job. They secrete a glycoprotein, and this glycoprotein is coating the primary oocyte. This glycoprotein has a name. It is called zona pellucida. Can we quickly draw a primary follicle? What is in the center of the primary follicle?
Primary oocyte.
Primary oocyte, nothing has changed, right? What is primary oocyte surrounded by?
stratified cuboidal.
Nope.
Flattened
Flat epithelial
follicular.
cells.
Nope.
Mesenchyme.
Nope.
Can you repeat the question?
Follicular cell.
Nope. Can't repeat the
Pellucida.
question. Not follicular cells. Yes. Zona pellucida. Primary oocytes are surrounded by zona pellucida. What is zona pellucida?
Glycoprotein.
Glycoprotein. Who makes this glycoprotein?
Granulosa cells.
Granulosa cells. Where are the granulosa cells?
Surrounding the lucida.
Surrounding zona pellucida. So on the top of the zona pellucida, you're going to find stratified cuboidal follicular cells, which we call now granulosa cells. This is a primary follicle. Is it clear? So the layers of the primary follicle are from the inner to the outer, primary oocytes, zona pellucida, granulosa cells. I'm missing here a stage because it wasn't in the book, but there is also a secondary follicle stage. So primary follicle becomes secondary follicle. It keeps on differentiating. What is the difference between primary and secondary follicle? Granulosa cells separate to form a little tiny cavity, little tiny antrum. If you see a little tiny cavity here, this is a secondary follicle. If you don't see a cavity in the granulosa cells, this is a primary follicle. 15 to 20 follicles can reach secondary follicle stage, but only one of them can reach the final stage of development, which is the tertiary follicle stage. What does the tertiary follicle consist of? You can see that this is the antrum. It got super, super big, right? So this is the cavity. It got super big. In the secondary, it isn't as big. In the primary, it is super big, as you can see here. What do we see here? This is the primary oocyte. Still. What is the primary oocyte surrounded by? Zona pellucida. Still. What are these cells? These are my granulosa cells. But granulosa cells, which directly surround your oocytes, these ones, they are known as the cumulus oophorus. These pictures are from the internet. Our university does not accept it. All of this is cumulus oophorus. It consists of the granulosa cells, right? These are also granulosa cells, but they're surrounding the cavity of the tertiary follicle. Only after ovulation, residual granulosa cells will create corona radiata, but we'll talk about it next class. For now, it means nothing to you. So this is simply cumulus oophorus surrounds your oocyte. Okay, can we quickly draw it? I go here. What does a primordial oocyte consist of?
A flat follicles.
Primordial follicle consists of the primary oocyte surrounded by the squamous or flat follicular cells. How many primordial follicles do you choose every month?
15 to 20.
15 to 20.
Out of 40,000, as you remember, starting from puberty. What makes them grow?
FSH.
They have a receptor right here for FSH. FSH binds to this follicle, forces this primordial to become primary. Let's talk about first stage of primary follicle. What are the differences?
The follicular cells become cuboidal.
But inside we still have primary oocyte. Am I right? So now follicular cells here are cuboidal, simple cuboidal. What is the second stage of primary follicle?
Stratified cuboidal.
But inside we still have
primary oocyte.
Primary oocyte. Here we have stratified cuboidal follicular cells. How do we call them?
Granulosa cells.
What do these granulosa cells do?
Secrete.
Secrete
Zona pellucida.
They secrete zona pellucida, which is surrounding your primary oocyte. So this red line right here, this is your zona pellucida. It is a glycoprotein, which is outside of the primary oocyte, but is below the granulosa, right? Primary oocyte becomes secondary oocyte. How? Secondary follicle. I apologize. Secondary follicle. Primary follicle becomes secondary follicle. Can we draw secondary follicle? What's happening? What is inside?
Antrum in the granulosa cells.
So hold on. Still we have primary oocyte. In the secondary follicle, we still have primary oocyte. What is this primary oocyte surrounded by?
Zona pellucida.
Always. Glycoprotein coat, zona, I'm going to put Z here, zona pellucida. What is here like this?
Granulosa cells.
There are lots of granulosa cells, but there's also a little tiny
Cavity.
space, cavity, antrum, whatever you want to call it. So as this space is present, this is a secondary follicle. In the primary, you can see there is no space. It's just zona pellucida surrounded by granulosa cells. Now, I want to get your attention to this area. Do you see this is a primordial follicle? This is a primary follicle. But do you see this yellow tissue? This yellow tissue is the connective tissue of the ovary, because of course, all of these follicles are sitting within the connective tissue of the ovary. The connective tissue of the ovary, which directly surrounds the follicles, has a name. This connective tissue is known as the theca folliculi. Theca folliculi. So if you imagine, if I take a, I don't know, this is like a piece of ovary, we have one follicle here, we have another follicle here, we have another follicle here. Is this theca folliculi? Follicle 1, follicle 2, follicle 3. Is this theca folliculi? It isn't. It's just a normal connective tissue of the ovary. How about if I put it here? Is this theca folliculi?
Yes.
Yes, because it's the connective tissue of the ovary which directly surrounds the follicle. Okay, so outside of it, you're going to see theca folliculi. Again, is this theca folliculi?
No.
No, because connective tissue, this is not anywhere near the follicles. If I say this connective tissue, is this theca folliculi?
Yeah.
Yeah, because it's next to the follicle. Do you see the difference? Theca folliculi, it's just the additional name for connective tissue, which is near the follicle. So if I go back, if I go all the way to this picture that we were drawing, what is outside of each of my follicle? What is outside of it?
Thicker.
Theca folliculi. What is theca folliculi? It is the connective tissue of the ovary. Okay, I'm going to stay here until the secondary follicle. Okay, we're gonna start with the tertiary follicle next class, which I think for most of you is tomorrow.